So is everyone here? Yeah. Okay, I'll start. Good morning and welcome all. I'm happy today to introduce to you all from Max Planck Institute for Marine Microbiology, Professor Nicole Dubilier, who's here today to talk about symbiosis. Professor Dubilier started her career at the University of Hamburg with a diploma in zoology, biochemistry, and microbiology. At the same university, she then went on to earn a PhD degree in marine biology with focus on marine oligochaetes and their adaptations to sulfide-rich sediments. After her PhD, Professor Dubilier continued with research on symbiosis in marine worms, first at Harvard University and then at Max Planck Institute for Marine Microbiology, where she now is the director of the institute as well as the head of the symbiosis department. And due to her astonishing contribution to science, Professor Dubilier has received several awards and honors, including Presidency of the International Society of Microbial Ecology, as well as the Leibniz Prize of the German Research Foundation, which is the highest research prize in Germany. And with that said, we are very excited to have you here with us today, Professor Dubilier, and we wish you a very warm welcome.
Thanks, Benjamin, for the kind introduction. And I'm excited about teaching my very first class in Sweden. Although it's online, who knows when the vaccines are finally available, maybe I'll be able to visit at some point. And as I just said, if there's a question for understanding, I'm fine with being interrupted. If it's more about a discussion, then maybe at the end of the lecture, we can discuss it. Okay, so I like to emphasize that chemosynthetic symbioses were discovered only 43 years ago, and many students obviously think this is ages ago, but many of the scientists that were doing research at the time that they were discovered are still around. It's a first generation discovery. And I also argue that this was the last major ecosystem on Earth to be discovered. Kind of like, that's it. This is the last place on Earth that we knew nothing about and then discovered thanks to our ability to take submersibles down 3,000 meters to the bottom of the seafloor. And they completely revolutionized our understanding of the energy sources that fuel primary productivity on Earth, because previous to their discovery, it was assumed that these types of large animal communities that you see here thriving in the darkness at 3,000 meters water depth could only be fueled by photosynthesis. And so we know that they are fueled by chemosynthesis, and I'll briefly go through that again, what that means. What is amazing is the biomass at these vents, the carbon amount per square meter. It's extremely high as that in tropical rainforests or coral reefs and kelp forests. And what is truly amazing is that the majority of this biomass is based on microbial animal symbioses. How do these symbioses work? The chemosynthetic symbionts use reduced inorganic compounds like sulfide to feed their hosts via CO2 fixation. And what I like to do is first present an image of what photosynthesis is, because that's very clear to many, where the light energy, the energy from the sun is used by chloroplasts, which are descendants of symbionts, of cyanobacteria. And the chloroplasts fix CO2 using the energy from the sun into biomass, and they feed their plant. And when we think about chemosynthetic symbioses, we like to call it dark energy, but it's actually chemical energy. It is the energy from the oxidation of reduced compounds like sulfide that is fueling the chemosynthetic symbionts, and the energy they gain from this process, they use to fix CO2 into biomass and feed their symbiotic hosts. Now, I'll get into that in a bit more later in my talk. It sounds very nice to, when I say they feed their hosts, actually their host eats them. They are digested by their host, they're fed on in most of the symbioses. So it's like farming or I like to sometimes compare it with hens on cradle. You make them nice and big and fat, the kids, and then you eat them. Except here, the kids are continuously wandering into the house of the witch. So what is really remarkable when we look back at the history of the discovery of these symbioses is we had to go to the bottom of the sea, 3,000 meters water depth at hydrothermal vents off the Galapagos Islands. It was geologists that were looking for hydrothermal vents to discover these communities. And we now know that they're widespread in the deep sea at certain sites where there is enough sulfide and enough methane to fuel them. Methane is another energy source at hydrothermal vents, cold seeps, and whale falls. And I'll come to that in a bit. But what I find ironic is that they are also very common in shallow water. So I like to say in our own backyards. In fact, in Germany, off our coast, on the island of Sylt, there are nematodes that have ectosymbiotic chemosynthetic bacteria. And for many decades, zoologists had often described animals that had a reduced gut or even had completely lost their mouth and gut. And it was assumed that they were living from organic compounds in their environment. But we now know that many, many, many of these hosts have chemosynthetic symbionts and that they are gaining their nutrition from these chemosynthetic symbionts. And so we collect these shallow water hosts that you see here in the Mediterranean, commonly in seagrass beds. They are very common in coral reef sediments, and you can actually also find them in sewage outfalls. Over the 40 years that we've been working on them, we used to assume, or no, we assumed based on the little knowledge that we had at the time that symbioses actually were not that common and that it was that their evolution only occurred rarely. But we now know that they are widespread and that these types of symbioses have evolved numerous times independently of each other in convergent evolution. And convergent evolution means in parallel evolution. So independently of each other, these symbioses have evolved. And we know that at least 15 bacterial groups have been able to establish these symbioses independently of each other. So it is not a single ancestral bacterial lineage, but many, many different lineages that did this in parallel. Same with the hosts. At least 2 protist groups and 9 animal groups have established these symbioses. And what this indicates when you look at this incredible diversity and you look at the fact that it has evolved in parallel numerous times is that there must be a strong selective advantage for both partners in forming symbioses. And I look forward to discussing that with you, what the advantages are when we go into our discussion session. These are just some of the hosts that form chemosynthetic symbioses and that we work on in our lab. The deep sea mussels is what I will be talking about for most of my talk, or for all of my talk after I get past the introduction today. One of our second major host groups here are gutless oligochaetes, little oligochaete worms. And in fact, the taxonomist who introduced them to me and who still goes on trips with us is from Sweden and the Göteborg University. And Krista Azeos, now retired, but used to be the only person that could identify these worms based on their morphology. Now, thanks to sequencing and MinIONs and everything you can take into the field, we can do a quick CO1 analysis. But he's still the only one who can keep them live and identify them for us. And the many other hosts here that we work on in my group. We just made this tree for a review that will soon be coming out in annual reviews in microbiology. And when Harald Gruber had finished making this tree, it really blew me away to see the diversity. We had always known that there are many, many lineages of bacteria. that have formed these symbioses. What you can see here are all the chemosynthetic symbionts that we know of today and their hosts. And you can see that many of them belong to the gamma proteobacteria here. You see a whole group here that I'll be talking about that are closely related to free-living chemosynthetic bacteria called Supo5. They're in the mussels that I'll be talking about. But we also know that epsilon, or now they're called Campylobacterota, horrible name, epsilon, I still call them epsilon bacteria, form these symbioses, and also alpha proteobacteria. Again, independently of each other, they have formed associations with hosts.
I have a question.
Go ahead.
So you said that there's so many, this symbiotic bond that's created with the chemotroph. It's only in invertebrates, right?
In protists as well.
Also in protists. And I was thinking about the invertebrates. Is it specific groups to invertebrates, or it's just all kind of invertebrates?
All kinds of invertebrates, and independently of each other. Let's see if I can actually go back. Well, you can't see it here, so the next time I'll have a tree of the host groups to show it more clearly. But flatworms, Paracatenula, are not related closely to nematodes. Nematodes are not closely related to mussels. So all of these associations have evolved independently. Within a given host group, for example, within the group of gutless oligochaetes, we then see 100 species at least that have descended from a single common ancestor. So once the association was established, often, let's say 100 million years ago, then it takes off within that group. We see what we call adaptive radiative evolution within that host group. But within the major lineages, it's evolved independently. Does that make sense?
Yes, thank you.
Okay, great, thanks. Okay. And by the way, exact same phenomena within the bacterial groups. So there, you can see my pointer, right? Moving around? Or?
Yes, we see it.
Okay, great. So for example, within this lineage, or let's take within the lineage of the Supo5, the free-living bacteria that are associated with mussels, there was one or two events where a free-living bacterium was able to establish an association. And from there, it then took off, because there seems to be something about once you're in, you're in. You've evolved the adaptations you need to associate with a host. And then from there, it skyrockets and takes off within that clade. And as I said at the beginning of the talk, we also now know that they're widespread, these types of symbioses in many different types of environments. One of my favorite environments are whale falls. When whales die and they sink to the bottom of the ocean, the meat on the whale is quickly eaten mainly by predators and sharks. So that's gone within about a few weeks to months. But the bones of the whales are full of oil, and sulfate-reducing bacteria degrade the oil. They produce sulfide, and we find the chemosynthetic symbionts in these environments. In fact, cows have been sunk, and cow bones, and brought down to the ocean. I think, in fact, I remember that in Sweden, we did some sampling once where we sank them in very shallow waters. And seagrass beds are full of chemosynthetic symbioses. And then one of my favorite sites for collecting the worms that we work on are coral reef environments, Great Barrier Reef, Caribbean, many beautiful sites for collecting them. Okay, we have a huge toolbox. We consider ourselves organismal biologists in that we are interested in the entire biology of these organisms. And so we use a wide array of tools to understand their biology, ecology, morphology. We collect them obviously at sea or in shallow water environments. Then we do a lot of imaging, and we I'll get to it. We haven't been able to cultivate any of the symbionts yet, not only we, the entire field, but we can keep our hosts in the lab, many of them, and so we can work with them in the lab. And then we use a ton of omic methods to look at the interactions between the hosts and the symbionts. For the rest of my talk now, I'm going to focus in more specifically on the research that we do on deep sea mussels to give you a flavor, a taste of some of the research that we're doing. And as I just mentioned, the symbionts remain as yet uncultivable. I'm convinced they can be cultivated. When students join my lab, they're often very psyched about trying to cultivate them, but obviously that's a project for a PhD where you shouldn't spend more. I always say, all right, you can invest a total of 6 weeks or maybe 8 to try this out. But after that, you do need to get a paper out within 3 years. And so that's in part also why it's been so difficult. But we've also tried a ton of stuff. Anyway, but we can keep the hosts in aquaria, even the deep sea mussels, if you collect them at relatively shallow water depths, 400 meters, 500, 600, then we've been able to keep them over many years in our aquarium. And so next I'm going to show you a video. It's a research cruise that we did in 2016. This is the Bremen campus where our institute is at. And we went on a German research vessel called the Meteor to the Mid-Atlantic Ridge, which is a mountain range at the bottom of the ocean in the middle of the Atlantic, where the ocean plates separating America and Africa and Europe are pulling apart. They grow about, well, let's not get into that. You just saw 4 vent sites where we went to with the German research vessel Meteor. And what you see here is a remote operated vehicle, the ROV. It's being let out to sea. It is flown. It's unmanned, an unmanned submersible. The pilots sit on the research vessel together with the scientists, and it's flown from a container lab in which the pilots can monitor the videos. And as you get closer to a hydrothermal vent, this is some of the first images you see a little bit of shimmering water. You start to see anemones. You start to see here these stalked barnacles. You start to see animals that normally you don't see at all in the deep sea. It's really barren. And then boom, it's like you've hit an oasis. You see these just beds and beds and beds of animals that are thriving there because they live in symbiosis with the chemosynthetic bacteria. Those are the primary producers. You see these absolutely gorgeous black smokers where the hydrothermal vent fluids are coming out at temperatures as high as, I believe the highest we've ever measured is 415 degrees Celsius. And what you're seeing here are metal sulfides. That's why it's black, because they're precipitating out when they hit the cold seawater. We take samples of the hot fluids because we want to know what are the energy sources for the chemosynthetic bacteria and the free-living chemosynthetic community, so that we know what are the sulfide concentrations, what are the methane concentrations. These Rimicara shrimp that thrive there have ectosymbionts on their gills. It's not really clear if they eat them. As you can see, the crabs, this is food chain, love to eat them. And what you see here are these mussel beds, and it's very similar to shallow water mussel beds with Mutilus, which is one of the well-known. They have an inhalant siphon with which they're bringing in the diffused fluids. What you see here is what it's like in a container van. We're working with the pilots closely. They're using the arm of the ROV. Here's one of the pilots and you see how small this little arm is that he's working with. They get shoulder and backaches just from manipulating them for so long. And this is one of the coolest experiments we're still analyzing in these little containers is RNA later for fixing the mussels. We wanted to fix them in the deep sea. So we asked the pilots to actually crack single mussels and place them within the RNA later so that we have them fixed at the sea floor. What you see here are mussels that we collected and displaced, moved away from the vent fluids, because we wanted to know what happens when they no longer have access to sulfide or methane. And the ROV is small. We can't always get all our instruments on it. So what you see here is an elevator that goes down separately from the ROV. And in the middle here, you see a very improvised virus collector where we were trying to collect the viruses in the deep sea and see what might infect these symbioses. The ROV is usually down for about 12 to 14 hours. The day begins at 8 in the morning with the dive. The ROV comes up at around 6 to 8 in the evening. This is the most critical step where we have to get the ROV on board. I've actually seen an ROV being that was lost at this step. It's a €2 million instrument, so you can imagine. And we're standing there, we're desperate to get our samples. We all race out as soon as we can to get the samples. They are in the front of the ROV and isolated. And here you see us racing to get these RNA later fixed muscles, which we compared to muscles that we hadn't fixed at the seafloor because we wanted to know what kind of difference is there between muscles that are fixed at the bottom of the ocean and those that aren't. Now you're going to see a little video where we zoom into what a muscle looks like. And you're looking at a muscle now in microcomputed tomography. And we are looking into a single muscle. And as it's being turned around in the computer tomography image here, we're going to zoom in the little threads or the byssus threads which it attaches to the surface it's on, and we're zooming through the animal. And you can see these layers of gill tissues, the brown stripe mass that you see here. And that is the symbiotic organism. It's greatly enlarged, and it's full of thousands and thousands of filaments, and I'll explain those in a second so that you can understand what these filaments are. And each single filament houses thousands and thousands of bacteria. So you see this incredible enlargement of the symbiont housing organ of the surface also within which they sit. This is a single cell in which they sit, and I'll show that in a little bit more detail. But we estimated that an 8-centimeter large muscle has about 50 million inhabitants. And we compared that to how many inhabitants Bremen has and the density of inhabitants per square meter. And you can see the density of the inhabitants in a muscle in comparison. So you are seeing a 40-fold enlargement there about 2 days on board in our buckets, not real aquaria. They are pretty much not happy campers.
And then when you like have them in an aquarium, are you like constantly feeding them with this?
Yes, I'll come to that.
The molecule.
Yes, let me show you that. That's going to be in about 2 or 3 slides.
Okay.
I'll show you what happens when they're in the aquaria.
Okay, thanks.
Okay, so now I'll just show you their morphology so you understand it better. What you're looking at here is the foot, which they use to move around. And these are these fantastically enlarged gill tissues. They're here in yellow again, and there's something like 10 to the 12 bacteria cells per host individual, but the diversity is extremely low. So we like to call these low diversity, high abundance microbiomes, kind of like a chemostat. That's what you can imagine what they're like, because there are only 2 bacterial species that occur in these muscles, a sulfur oxidizer and a methane oxidizer. And so this is what it looks like when you get into the morphology. And the reason I'm explaining this to you is so that you understand the adaptations on the host and the symbiont side to living inside an animal. It's not that easy for a bacteria to live inside an animal. And so what you see here are these filaments, and you can compare the morphology of a muscle gill to a radiator. And what you see here are in the radiator are about 8 different fins. Those fins are what we're calling a filament in a gill. And very similar to a radiator, you've got the inside of the radiator, which is where the hot water is in. Well, that's the inside of the muscle filament, which is the body fluids of the muscle. And then on the outside of the radiator, you've got the air that is being circulated through the radiator to get warmed up. And it's very similar here in the filaments, where you've got the seawater that the mussel is pumping in that is being streamed through these filaments. And what you're going to see here is a cross section through two of the filaments, so two of these radiator elements. And we're looking at the outside here, where the seawater is constantly, the black here is where the seawater is streaming through. And inside is where the mussel tissues or fluids are. And each single, this is a single host cell that is outlined here, a single host cell here, a single host side, is chock full of bacterial symbionts. And we're using a method called fluorescent in situ hybridization with probes that are specific to the sulfur oxidizer and the methane oxidizer so that we can see where they are. And another fascinating thing that you can see in this image is that there are host cells that are free of symbionts. These are at the outer edges of the mussel where it's has cilia that it is streaming in the water with. And we think because the energy demands are so high in this area, it says, no, I'm not letting you in these cells. But right next to it are the cells that are full of symbiotic bacteria in which then the animal allows, let's just use those images, allows the cells to be colonized. Now what you're seeing is an electron micrograph. So we're increasing the resolution as we go in further of a single host cell within a filament, a single bacteriocyte. And what you can see is that the sulfur oxidizer, the smaller bugs here, and the larger bugs here, the methane oxidizers, are sitting inside the host cell. There's a very, very cool, very, very new story we have here, which I don't have time to talk about, but it looks like they actually might be pores connecting them to the outside here, but I won't go into that now. Okay, so this is what a non-symbiotic, if you're now looking from the front of a mussel and you're going through a non-symbiotic blue mussel, Middleus, that you can find all along the Atlantic and the Mediterranean. These are what the gills look like if you're looking from the front of the mussel. And this is what a gill cell looks like. And now compare it to the fattened, just super chunk hypertrophied gills of a symbiotic mussel. And this is a single gill cell that you see here that has become greatly engorged and huge and large because it is hosting the symbiotic bacteria. And the way the symbiosis works is that this is a single filament. This is the inside of the mussel. This is where the body fluids are. And these are the single bacteriocytes. They're full of the symbiotic bacteria here. And these black blobs here are not poor electron micrographs. They are what is called phagolysosomes, which are well known from pathogenic infections with which animals get rid of the waste in their body, but also eat bacteria. So they use it to eat infectious bacteria, but to get also rid of mitochondria that aren't functioning. And so it's kind of a conveyor belt with the symbionts multiplying, growing here in the top end, the apical end of the animal host, and being eaten by these phagolysosomes at the bottom end. Okay, so you asked about what is it like when we keep them in aquaria, and let me check the time because we're reaching 9:30. So I'm going to probably not go into all the metagenomic analyses we do, but rather let you ask questions as when I have finished this part. What we know, we collected mussels in the Gulf of Mexico in 2014, and then we kept them in aquaria at Kiel at the Guiomar. Frank Meltzner from the Guiomar helped us with this. And we kept them under steady methane. These are mussels that only have methane oxidizers. And we kept them in the aquaria for, oops, don't want to give that away yet. For 1 year at 10 micromolar methane, which is quite a high methane concentration, and constant flux. So in the aquaria, we were bubbling it with methane continuously and with oxygen to make sure they get enough oxygen. And then the reason we amped it up to 40 micromolars, I'll show you in a second, because this is what happened to the mussels. This is what they look like, a single host cell, a bacteriocyte, when you collect them freshly from the field with all the methane oxidizing bacteria here, chock full, some are dividing. This is what the poor mussels look like after 1 year in Aquaria at 10 micromolar methane. Is not a single symbiont is visible. And you'll see another interesting actually adaptation, which is on the host side, which is that it regains its microvilli. So the host cell looks very different. And then after we amped the concentrations up to 40 micromolar methane, they started to regain their symbionts. So what you can see here is, and by the way, the scale bar is the same for all three micrographs that you see here. So we're looking at the same size of the host cell. What you can see is that they have started to regain their symbionts. So this is very similar to something that we know from coral symbioses, where depending on the conditions, the corals can actually bleach, lose their symbionts, and regain them. I spent a lot of time, I was slower than I normally am, but that's fine because I like to explain things more carefully to when I'm teaching in a class. In the second half of my talk, I would have concentrated on all the omic work that we're doing and shown you some of what's in there. But I think in the interest of letting you ask lots of questions, I'll end here and go, let me see if I can go to my last slide, where at least you'll see the wonderful people that have been doing the work here. Which is this slide. At our recent retreat, we had our first online retreat, and the work that I've been showing you has been done by a fantastic group of students and postdocs in my lab and has been supported by generous funds. And I'll thank you for your attention, and I will stop sharing my screen.